In this video, we look at a part of drug development called target selection. You will remember that target selection is the first part of the drug discovery pipeline, and the aim is to find a target for a drug and then to find drugs that act on that target. So finding a target then means finding something which, if a drug acts on it in some way, it will have the therapeutic outcome that you're looking for. So how do we find targets? One thing you can do is to use physiological knowledge. So for example, let's say you're trying to find a drug to treat blood pressure, and you happen to know from your physiological knowledge that angiotensin II causes an increase in blood pressure because it acts on angiotensin receptors. In which case, the rational thing to do would be to choose AT1 receptors, these are receptors to angiotensin II, and choose those receptors as your target. Because angiotensin II acting on these receptors causes an increase in blood pressure, you would be looking for chemicals which antagonize this action. That is, they are antagonists at angiotensin II receptors. But as you can see from our diagram here, there are actually several other targets. We've looked at angiotensin receptors. We could also look at beta-adrenergic receptors. We know, for example, that beta-1 adrenergic receptors increase the rate and force of contraction, and that of course increases blood pressure. So why not choose beta-1 receptors as your target, and again look for antagonists acting on this receptor. Once again, we also know that angiotensin converting enzyme stimulates the conversion of angiotensin I to angiotensin II. That's this part here. So what we could do is to choose angiotensin converting enzyme as a target and use peptidases which inhibit this enzyme. So then this is using your physiological knowledge to select a target based on what you know that that target does. And given what the natural ligand does, you can then choose what sort of action you're looking for. So this is choosing a target based on physiology or knowledge. But you can also expand that slightly by looking at targets related to ones that you already know about. What I mean is that if you know a target, then you can also find other proteins that interact with that target and select one of those as a target as well. So here's a situation in which we know that here we have the gene for angiotensin receptor 2, and from databases you can find out that there are other proteins which physically interact with your target protein. Perhaps these proteins are essential for your target to work. So rather than just approach the angiotensin receptor, you could maybe try to select one of these other genes as a target as well. There are many new technologies that open up new targets like this. And in general, we use the word omics to refer to techniques which use techniques that cover a whole area of a field. So for example, proteomics, which is what this is all about, is the study of all the proteins. Transcriptomics is studying all the messenger RNA. Another modern way of choosing a target is to use computer modeling. Computer modeling makes a detailed physical model of your target and then tries to work out which sort of molecules would fit in by simulating the movements of all the individual atoms and looking for what we call accessible targets, that is, bits of the molecule where the drug would land and predict using things like machine learning and what we know about the properties of the pocket to see what sort of drugs would work. This can really speed up the choice of a target. So what are the most common targets that we currently use? Now this looks like a complicated figure, but it contains a lot of information about drugs and their targets. So what we're plotting up here is, in this one, a pie chart of the proportion of human protein drug targets in the major families. These are G protein coupled receptors, these are ion channels, kinases, nuclear receptors and other. And as you can see, the most common targets are actually ion channels. Now I'm going to make things a little bit complicated now with what appears like a contradiction, because if we look at the proportion of small molecule drugs targeting each receptor family, we find that G protein receptors have the most drugs directed to them. So most drugs target G protein coupled receptors. How do we reconcile these two together? Well, if you think about it, you can have 3 drugs against 3 ion channels. So in this case, most targets are ion channels. And here we've got 5 drugs, but they're all shared between just 2 G protein coupled receptors. So that's how we can reconcile these two statements. But it still remains that in proportion to the genome, ion channels are the most common type of target. We also use genomics now to help find targets in many, many different ways. And it's quite fun to look up how drugs are being discovered today, because it's rather clever how they use so many different techniques that are available in our toolkit today. Here's an example I thought you might find interesting. This uses a method called short interfering RNA. It's a form of RNA interference. And what you do is you take a lot of cells and then you transfect them with this construct here called a short hairpin RNA. And these are randomly generated against all the genes in the genome. So you infect them with this special construct which will knock down just one gene in your cell. So then what you do is you assay them and you find out which cells are being affected. And then what you can do is you can take those cells, and here's a trick, you add a barcode in this RNA so that once you take that cell that responds to, say for example you've got cells here which are cancerous, and you might find ones which don't grow anymore because in their case they just happen to get the short hairpin RNA which has knocked down one particular gene which is necessary for the tumour growth. You obviously want to know what that gene is, because that would make a great target, wouldn't it? So once you've infected them all randomly, so you pick out cells which are growing slowly, how do you find out which was the one they were infected with? Well, what you do is you have a barcode. And then what that does, it gives you a whole list of genes whose knockdown affects the phenotype or the disease that you're looking for. And that's just a way of giving you a whole bunch of new targets. So really what you're doing is you're randomly knocking down one gene in the whole genome and then seeing whether that cures the disease. And then all you've got to do is say, well, I know that if I knock down this gene, I stop this disease progressing, maybe this would make a good phenotype. So where are most drugs targeted to? Well, this diagram is one of those which looks quite horrendous at first, but actually very neatly presents an awful lot of very useful information. So what you've got here is you see these letters? These represent the target system. So A is the alimentary tract, or the gut if you like. M is the muscle system. N is the nervous system. And then you've got the most common targets here. And then for each one, you've got the approval year of the drugs. So if you see a big stack like this, that means a lot of drugs are directed against that target. And if you see a lot of red, you know that these are more recent ones. Now you notice there are not many antiparasitics and not many new drugs there, whereas for anti-neoplastic and immunomodulating agents, so this is for cancer and immune disorders, we can see a lot of new drugs being developed here. And then if you look on this diagram, you've got a similar sort of thing. You've got the types of receptors: G protein coupled receptor, nuclear receptor, ion channel and kinase. This confirms what we saw before. There's a lot of GPCR drugs, but look at kinase. We can see that there aren't many there, drugs aimed at kinases, but there's a big red bit, which means that this is a future, this is a new area which is developing very rapidly. Ion channels have had their day because there are very few new drugs over the last 4 years. G protein coupled receptors are continuing to grow. So this gives you sort of indication of the direction of the field.